So this is the presentation for now, so the importance of terminal sterilization in the pharmaceutical industry. I will start with that nice picture. Would you take that risk? Personally, I wouldn't, as I'm definitely not a great skier. But the real question is, do you find that risk as acceptable? Do you consider that taking that risk is nice for you? I go too quick. A snowboarder is considering 20% risk of falling or broken bone during a season. And for him, that's acceptable because he says that he gets some rewards doing these sports. So do we accept that risk or not? That's the question. And we compare it, I will compare it with the sterilization, because sterilization is always a risk. You can never reduce the risk to zero, but there are ways to reduce that risk as much as possible. So we will see together how we can do that. Microbes are everywhere. So a single can cause infection or even death if it penetrates in unprotected zones. So really here, the question is, how can we make sure that we eliminate them? But in reality, we cannot totally eliminate. We can just reduce their population in a way that it does not create any problems in the patient. Sterilization and sterility, that's all about that. That's how to make sure that you do not let the microbes entering into the unprotected zones. And it's difficult to do. Definitely, we see some numbers here published from FDA. So we have nearly 1 recall event registered in the FDA website about problems related to lack of sterility assurance. So nearly 1 event per day. That's really a huge number, considering all the impact it can have on your company reputation, the additional costs behind that, and of course also the problem for product availability and the patients at the end. But what does this mean, a lack of sterility assurance? What is the meaning of assurance of sterility? This is the sterility assurance, that's the probability of a surviving organism to be in a sterile product. In fact, you have a sterile product, you can define it when you have a risk or a probability lower than 1 in a million to have 1 unsterile product. So that's really, and I'm going too quickly. That's really the official definition for a sterile product. So yeah, being said like that, that's quite easy, but not easy to obtain in a practical way. Especially because absolute sterility can never be totally guaranteed, because you will never be able to test 100% of your batch. That's not physically possible, otherwise you will have no product anymore. And even if you can test all of your products, you have no assurance that your test will be able to detect any microorganisms. So that's something to keep in mind all the time. You will never be able to totally guarantee sterility of your product. And that's probably a unique situation where it's required by law, but you have no way of totally proving it. And that's why it's so important to select your sterilization methods in the right way. That's why it's so critical to select it the right way. To produce a sterile product, you have in fact 2 totally different ways. The first one is aseptic assembly, and the second one is terminal sterilization, and my pointer is not going well. Terminal sterilization. And I take again the analogy of ski slope to illustrate these two possibilities. Always when you are a decision maker, you have to choose the more adequate way to sterilize your product. It can be aseptic or it can be terminal sterilization. And I let you decide which slope color is representing which technique of sterilization, because of course each of your situation is unique. But anyway, when you are at the top of the mountain, you will do your decision based on risk and considering all your environment to get at the end a sterile product. If we do compare the two methods of sterilization, so we will do that together, just a big picture to know a bit more about these two technologies. So for aseptic assembly, that's really a question of maintaining sterility. So there you have different products already sterilized by different methods, and you assemble them to get at the end an assembled sterile product. Everything has been previously sterilized. For the terminal sterilization, the way to do that is totally different, because there you have really an effective exposure of your terminals of your product. So you have the drug, the container, closure, etc., and you expose it to a physical agent for a specific treatment. There it means at the end for both you have a sterile product, but you see the way to achieve it is totally different. Let's go a bit deeper into the terminal sterilization pathway. Terminal sterilization is the recommended method by the authorities. So especially, or I took the example of European pharmacopoeia, where it says that wherever possible, a process in which product can be sterilized in its final container is chosen. So that's really the recommended method. That's the first one to consider. But why? Why is it so pushed by the authorities? That's because terminal sterilization is really based on real validation. I mean, you have a linear relationship between your product bioburden, so the natural contamination of your product, and the treatment applied. So it means that you can predict the extent of treatment to achieve your sterility assurance level. So this is really the basic principle of the terminal sterilization. It's based on calculation. Here I illustrated with this curve. So you start with a population of 1 million if you are using a biological indicator. You do your sterilization process, and you can predefine how many doses or how many time you need to achieve the sterility assurance level of 10 to the -6. So that's clear, that's theory, and that's really well known. Also, you compare it to the natural product bioburden, which is usually lower compared to the witness to the biological indicators you are making your validation. So it means that you are adding some safety margin by your calculations. So really, you can predict how much dose or time you need to achieve that level of sterility assurance. So that's the basic of terminal sterilization, and you should keep in mind to compare it to aseptic assembly. What are the advantages and disadvantages of that method? And the list is not exhaustive, but it's just to give you an idea and a comparison of the two. So terminal sterilization, if you go to a facility making that terminal sterilization, it's done in a standard industrial environment. So for those of you who are working in a clean zone, in a clean environment, that's nothing to compare. That's really like a normal facility where you do not need really to have all these additional controls required in a clean room environment. That's also a fully automated process, so it means that your parameters are fixed and validated, and you can reproduce it with your equipment. So that's really validated and reproducible. For terminal sterilization, you also have the possibility to group your product. So it means that you can sterilize together products that are totally different, but on the terminal sterilization point of view, you can group them and then only have one sterilization run instead of different batches. That can be a huge advantage for that technology. There are limited QC tests required. Probably you're not aware about that, but ISO does not require to do a product sterility test if you're using terminal sterilization, because it does not bring any real added value. It's not necessary. Also, the volumes can be very large in our facilities. For example, we have chambers starting from 1 euro pallet to 32 pallets, so which is huge. That's like a full truck. So also there you can play or have different possibilities for volume treatment. For the disadvantages, of course, material compatibility should be checked. That's the first thing you will assess during your selection of methods. And you will also have to check the impact on your product. It can have some residuals on it if you are using ATO residuals, or it can have some extractable and leachables, and there my colleagues are experts in there to assess that part. But that's something that could happen if you are exposing your product to a sterilizing agent. Also, that's typically a process that is subcontracted, so then your turn times could be a little bit longer compared to in-house process. What about aseptic assembly? So aseptic assembly, so remember that's really the other way to get some sterile drug products, and there you cannot calculate your SAL, so you do not have the curve I explained before. It does not exist because there is no killing kinetic in such a process. That's totally a different approach. And there it relies, your process is made of different steps. For sure, if you are in that pharmaceutical aseptic processing, if you are familiar with that, you know how much complicated it is to get that sterile product. And then you have all these related factors like human factor, the equipment, all the environmental controls and things like that are very, that's hard to control of these elements to maintain the sterility of your product. Also, the way to measure the efficiency of your product is done through MFT, so media field test, and that's process simulation. So that's not the process itself. That's you make a media passing through your process, and that's the way you estimate how much contamination you could potentially have on your product. The contaminated or the potential contaminated products are expressed in a default rate called PNSU, so probability of non-sterile units. And there, a failure of 1 in 1,000 samples is admitted. So put it on the balance compared to the 1 in 1 million. So nothing to compare there. And that's offline measurement, because you do your MFT before or after your process, but not necessarily or during the process itself. Again, a short comparison or with the advantages and disadvantages of the different methods. For the advantages, as you are using already sterilized components, it means that you can use different sterilization methods for your different components. So it can be optimized for each of them, which can be an advantage. That's also usually an in-house process. And there also we see more and more sophisticated equipments like wraps and blow-fill-seal technology, which is also good for improving the process. And that's definitely a well-known technology in the pharma industry. That's very popular. For the disadvantages, as we said before, it relies on different factors, like human factor, environment, and the different utilities. So of course you need to control and make sure that you have all of these in the proper control. Consequently, you need to do a lot of quality control tests, like product sterility tests, but also all your environmental testing, all the trainings of your personnel, all these kind of things have to be very well managed in the aseptic assembly world. And consequently, the costs of manufacturing are also very high. If we consider now, if we take now the lens for risk assessment, let's see what are the risks. And here the risk is having a non-sterile consider all the elements. So here we will use ICHQ10, which is using the basics for this is the scientific knowledge for the assessments. And you should balance your efforts compared to the risk level you will assess. We will go through the 3 factors that are usually used for risk rating, which are probability, detectability, and severity. First, detectability. So if we go through the aseptic assembly, how easily can we detect an unsterile product in aseptic assembly? So that's offline control. So consequently, that's an indirect control. You do product sterility tests, but we know it's not 100%, it's not showing 100% of your contamination. And it's based on a sampling frequency. So based on the USP, for example, if you have a batch of 100, you need to test 10% of your batch, which means that it could happen that the other 90% are contaminated. So you will never know that. For the terminal sterilization, it's another story, because there you have really fully control online and direct. And the frequency for the testing is total, because you are really checking your process itself. So here we clearly see that the detectability for aseptic assembly is much more lower in the aseptic assembly compared to terminal sterilization. For the probability, so we talked about that a bit before, but that's estimated with a p-NSU, so the accepted rate is 1 in 1,000 samples, and that's done offline. For terminal sterilization, As we say, that's one in a population of 1 million, and it's quite frequent to have much lower SAL compared to that one. So you have usually 1 in 100 million samples is something quite frequent. So again, we see that the probability is definitely lower in terminal sterilization compared to the aseptic assembly. What about severity? Severity, yeah, here that's quite simple because I would say for both, that's the same. We are considering the patient, so if we get an unsterile product, of course, that's equal for aseptic assembly and terminal sterilization. So if we consider the combination of these three factors, we have the risk, and how can we control the risk? Definitely using terminal sterilization is a way to reduce your risk to control it. What are the benefits you can expect beyond decreased health risk? So I don't know if you would have some ideas about that. On our side, we made our own survey and we asked customers, so we asked a third-party company to do that for us, and we asked to many pharma manufacturers what other benefits they could have if they are using terminal sterilization. And these are their answers. So it maintains product availability because you have less risks of recalls. It minimizes liabilities and also preserving corporate reputations. Of course, if you have less risk of recall, you have less risk of problems on the market. Also, when you go to the authority with a dossier for a registration of a new medicine, if you can tell the authority that it will be terminally sterilized, that's quicker. Your dossier will be quicker approved, which is also very nice to have. They also told us that they realized some cost benefits there, and also technical benefits. And there, that's something we have from experience. We had some nice projects where the drug products were better after being exposed, for example, to gamma. So they are more stable after exposure compared to the non-exposed ones, so which is also a nice and sometimes even unexpected benefit from sterilization. Here's a PDA survey, so it's 2 years old now, but there the manufacturers have been asked about the method they are using, and here we see that 75% are using aseptic assembly, but even so, amongst them, 30% could use terminal sterilization. So based on what I said before, this is quite surprising, so we can ask the question why. I do not have the answer, but I know that this is a change for pharma customers to consider terminal sterilization. So they are more at ease with aseptic assembly. They have a lot of experts there for sure, and it takes time to move and to consider other technologies. So, but anyway, 30%, that's quite huge. That's also in the actuality because the EMA guideline, the EMA published a recent guideline, so it started into effect the 1st of October this year, where really they explain how to select your sterilization methods. And again, here we clearly see that the blue colors box there are related to terminal sterilization. And the first one you should consider, that's heat sterilization. If it does not work, you can go for radiation, and if your product is not able to support that, you can go for other terminal sterilization. But the order or the sequence is very clear there, that you have to go first to terminal sterilization before considering filtration and aseptic, or even aseptic processing alone. And you should justify your choice. So it's not enough to say, oh no, it's not valid. You have to show that you made some tests to demonstrate that terminal sterilization was not feasible, was not possible. And yeah, it's recommended to use a structured approach to do that. When do you have to think about sterilization? I would say the sooner the better. And you see here the whole process of product development. But I have frequent projects where customers are coming to us and asking or thinking about sterilization where they are already making some clinical trials, which is usually too late because the packaging is already defined and it's already late to change something. So the best moment to think about sterilization is definitely during the early development phase. So don't hesitate to come early to see us and to discuss with us. We can surely guide you for the best options. Prior to making your choice, you have to see if you can adapt something to your product. Maybe you can change your formula, adapt some excipients that could resist to exposure, for example, or maybe you can adapt your container, or maybe you can select other components. So that's your the possibilities you have on your manufacturing side. After that, you should consider and talk to your manufacturer subcontractor to see if the sterilization process itself cannot be optimized, because there are different options there. And we will briefly see some examples. Possibilities for radiation mitigation. So a first one could be to use lower dose. So probably a lot of you are familiar with the 25 VD max with 25 kilograms. There are other possibilities where you can have lower dose. We also have projects with using deep freeze or inert atmosphere. Also, most of people think that it's not possible to irradiate aqueous solutions, which is not true. We have good examples there. So these are some possibilities for radiation. Ethylene oxide, it's also possible for drug substances. We also have some nice projects there with different APIs also. And there, it's not necessarily that you have to use the well-known overkill method, which is very big cycles. There you can have some optimized cycles, really limiting the residuals and impact on your product. We can have also cycles not in deep vacuum to preserve the packaging, low EO concentration, low temperature, and things like that. So this is possible with ethylene oxide sterilization. Other possibilities, noxylizer. That's a new method we are testing in one of our facilities, giving promising results there also. And that's a surface sterilization process, so the ingress is limited, and it's very interesting, for example, in pre-filled syringes. The way the process is done is also nice for pharmaceutical products because it's running between 10 and 30 degrees, and the residuals are also much lower compared to what you have in ethylene oxide sterilization. So yeah, the residuals are not cytotoxic. So yeah, promising technology also. And also something to consider, which is not taken into consideration sometimes, that's to use a higher SAL compared to 10 to the -6, because there, even if you are using 10 to the -4, you're safer compared to aseptic assembly. So there it needs to be approved by the authorities, but that's a possibility. And that's something you can probably defend if you are in a situation where aseptic assembly is also possible. And there I refer to the ISO guidance there, which is a very nice support if you want to go to that way. Here's some examples of products we are sterilizing in our different facilities. So with radiation, we have, for example, nanosuspensions or lyophilized products. Many APIs or dry powders are sterilized with the radiation, so it can be gamma or E-beam. With ethylene oxide, we also have different projects with different customers sterilizing APIs. And also nice applications for external sterilization like pre-filled syringes or applicators or even active implants. So that's something we see more and more. Nitrogen dioxide noxylizer can also be used especially for pre-filled syringes. That's it. So to summarize, could you control your risk and consider terminal sterilization first? So yeah, of course, I stay at your disposal at the booth if you want to discuss it further. Thank you very much.